Okay, here's the second figure that we're covering. So this figure in chapter 20 and the textbook section that describes it. And this figure also takes advantage of what we learned just in chapter 16, and that is we're going to synthesize DNA in a test tube, and we're going to have to put in the test tube DNA polymerase, and we're going to have to put in the test tube the things DNA polymerase needs to get its work done. We just learned about that in the method from the previous mini lecture on DNA sequencing. And in DNA sequencing, we do that because we want to read the sequence. We want to read the sequence of letters in the DNA, A, G, C, T. And so if we tagged each one with a special color, we were able to identify those colors and thereby read the sequence. Okay, so this figure, this next figure in the book on chapter 20 is a different method. It's called PCR, which stands for polymerase chain reaction, and it's done for a different reason. It's done to get more DNA. And there are many applications where scientists want to get more DNA. They refer that getting more DNA as DNA amplification. So we're going to copy the DNA repeatedly, and we're going to amplify it and get lots and lots of it. Biochemistry historically was involved, featured a lot of purification, and so instead of purifying specific DNA, we're going to copy specific DNA. And the way to think about that is, let's say we want to study your insulin gene. Well, traditionally we would isolate it away from all the rest of your genes. And now with PCR, instead of isolating it away from the other genes, what's done is the insulin gene is copied millions of times, so that it ends up being almost all of what's present. The other DNA is still present, but there's just a superabundance of the gene of interest, the DNA of interest that is studied. And there are lots and lots of applications. So for example, in crime scene forensics, if there's a drop of blood, that's enough DNA to amplify and study and analyze to determine who the criminal was, like that. Or you see, like in Jurassic Park stories, there's some DNA preserved in some ancient source, and scientists amplify that DNA. Then just in manipulating DNA, like we've talked about adding a promoter from one gene and transcription unit from another gene, though often the way those experiments are done is to amplify the pieces that the scientists want and then to put them together. So there are lots and lots of applications, and as a result, the guy that came up with the idea, you wouldn't really call him a discoverer but an inventor, Kerry Mullis, won the Nobel Prize, and he's a pretty crazy California surfer, so it's kind of funny on the web to read about him and his lifestyle. But he was working at a company called PerkinElmer in San Francisco when he came up with the idea of PCR. And so here it is in the textbook. And it just shows here that what's added to the test tube is the DNA polymerase, and then what it needs to do its job. And we need to add a template, something for it to copy. We need to add primers. We need to have the nucleotides. Now, we get to add the template. So if we're studying a human gene, we can add some human DNA and study it. And then here's the kind of key for PCR is we get to pick the primers in this case. So back in chapter 16, RNA primase was making primers for DNA replication in S phase. But in this experiment, we're in a test tube with no cells, and we just get to add what we want, and we can add primers that we want to be directing the DNA polymerase to the gene of interest. So here's a long piece of DNA, and here's the insulin gene. And we can add a primer here and here, and consequently just focus the DNA polymerase's activity between this region so that we'll get products out, and we'll get many, many products of that same length. And so we can add a big mixture of DNA and focus by priming in specific regions, focus on this specific region. And the process is like photocopying repeatedly. So normally there are 20 to 40 cycles of DNA replication. And the book shows a couple of cycles here. So during the cycles, what are done is first the DNA is unzipped, and that's called denaturation. Denaturation in a cell, denaturation or unzipping is done by proteins. In the test tube, the scientists do it with heat. So in the same way that you can unzip, unfold a three-dimensional structure of a protein by heating it and irreversibly denaturing it, you can also do that with DNA. The DNA hydrogen bonds between the nucleotides can be unzipped, separated, and so it's heated and then it's cooled. And when it's cooling, the primers can now stick, and the primers stick in these locations. And then after cooling it, the temperature is warmed a little bit because it's the optimum temperature for DNA polymerase to copy. So DNA polymerase copies during the extension time, and then that process is repeated. So we do a cycle, we get 2 copies. When we do another cycle, we got 4 copies. Do another cycle, we've got 8 copies. So we've done what we have every time, and pretty quickly we get just what we want, the DNA between the 2 primers, and lots of it. And that's all there is to this, but you want to make a separate figure and a separate mini lecture because of all the different applications there are for doing PCR. The last image is just to show you it being done because it's just kind of curious to see. It's just a simple instrument that is really just a heating and cooling block that you can program to heat and cool at specific times and numbers of times. And then the test tubes that are used are really tiny, simply because it's expensive ingredients that go in there, so only small volumes are used. And then also a small tube is easier to heat and cool quickly, and it's got to do that, like I said, 20 to 40 times. And that's PCR.